Hello and welcome to the latest edition of my blog. Today we're going to be talking about the plain language summary of my latest paper and what can we learn about antimicrobial resistance from hospitals for people and animals. So this is a plain language summary of a paper that we wrote, so that's myself and colleagues across University College London and the Royal Veterinary College, which describes how we compared two different metagenomic data sets, one from environmental samples taken in a human hospital ward and one from environmental samples taken from a companion animal veterinary practice. In this paper, we specifically looked at the use of metagenomics, which is also known as the study of multiple different organisms within a single sample using sequencing. At the Centre for Clinical Microbiology at University College London, we naturally work very closely with the infectious diseases team in the local human hospitals, and scientists and clinicians of both the human and animal variety are becoming more and more aware that we need to link up and share our knowledge, as no sphere exists in isolation, and the linking of animal, human, and environmental research is known as One Health, and it's becoming an increasingly important concept. As part of our work with Pandora IDNet, the big consortium that recently finished, CCM also works with the Royal Veterinary College and other more animal-focused sites to undertake One Health projects and provide capacity development for others to do the same. And what we did here, and this is a plain language summary of the research that we did, and if you want a link to the original paper, you can find it in the accompanying blog page. So the background. Antimicrobial resistance, or AMR, is when bacteria evolve so that the antibiotics that we used to use to treat them no longer work. And one of the biggest challenges facing modern medicine is this resistance. Hospitals are particularly important in this story because they bring together vulnerable patients, frequent antibiotic use, and lots of opportunities for those microbes to spread. But hospitals don't exist in isolation. Human health care and veterinary care are closely connected. Of course, many human patients will have pets at home, and all pets will have a household of various different guardians or owners. Bacteria can move between us, our animals, and the surrounding environments. In our study, we wanted to better understand how these systems are linked. Specifically, we compared the microbes and the antimicrobial resistance genes present in these two different healthcare settings in London: a human tertiary or very large hospital and a companion animal or veterinary hospital. Instead of looking only at individual bacterial isolates, so individual sort of E. coli or Klebsiella, we used metagenomic sequencing. Now, this approach analyzes all of the genetic material found in a sample at once, allowing us to capture a much broader and more realistic picture of the microbial communities, the resistance genes that they carry, and mobile DNA elements such as plasmids that can spread these resistance genes. Why is it important to compare human and veterinary hospitals? Human and animal health, they're both deeply connected. One Health recognises that people, animals, and the environment share microbes, resistance genes, and other DNA, and veterinary hospitals are also hotspots for antimicrobial use and resistant bacteria, but they're studied much less often than human hospitals. By directly comparing these two environments, we wanted to answer several key questions. Are the microbial communities similar or different in human and animal hospitals? And so that is the microbiome. Do they carry similar antibiotic resistance genes? So this is known as the resistome. Are there shared genetic elements such as plasmids that could spread resistance between these settings? So this is known as the plasmidome or the mobilome. And what might this tell us about how resistance moves between people, animals, and the environment? What did we do in this study? We collected environmental samples from both the human and the animal hospitals, and these included surface swabs, so these are things like door handles, floors, and toilet bowls. And we also collected wastewater samples, which capture much more complex microbial communities, so think sink U-bend water and toilet water. We extracted all of the DNA from these samples, so this included everything: bacteria, viruses, fungi, human, animal, whatever was in it at the time of collection, and then we sequenced that DNA. In total, we analysed 36 different samples from the human hospital and 48 from the veterinary hospital. Once we got this sequence data, We analysed it to try and identify the microbiome, so what bacteria and other microbes were present, the resistome, which antibiotic genes, resistance genes are present, and then the plasmidome or the mobilome, so looking at the mobile genetic elements that might carry those resistance genes between species. Moving on to what did we find? So the first thing was that the different environments had distinct microbial communities. One of the clearest findings was that the two hospital environments had quite different microbiomes. This was, I don't know, both surprising and not surprising. Of course, the patients, humans and animals, the treatments and the physical settings are quite different. But people and animals also share a lot of environments where each could pick up microbiomes from the other. So think your very lick happy dog that you might have at home. And these are just two individual hospitals, of course, but it does confirm that each hospital type, in this study anyway, had its own ecological sort of signature. Despite these differences, there were also both overlapping groups of bacteria, um, most commonly Salmonella and Campylobacter, both of which are zoonotic, so means they can be passed between humans and animals, suggesting that there are some microbes that are well adapted to healthcare environments in general. The second thing we found was that both hospitals contained a wide range of resistance genes. We found that antibiotic resistance genes were widespread across both settings. These included genes associated with resistance to commonly used antibiotics, that's in both human and veterinary care. This highlighted a really important point that AMR, or antimicrobial resistance, is not confined to human hospitals alone. Veterinary hospitals can also harbour diverse and complex resistomes, reflecting antibiotic use and microbial exchange in animal healthcare. Thirdly, mobile genetic elements played a key role. A particularly important aspect of AMR is not just which genes are present, but how easily they can spread. Many resistance genes are carried on plasmids, which are small pieces of DNA that can move between bacteria and sometimes fungi, and frequently carry these AMR genes. Our analysis showed that these plasmids and other mobile genetics elements are present in both hospital environments, creating opportunities for resistant genes to transfer between different bacterial species. This mobility is one of the reasons that AMR can spread so quickly and unpredictably. 4. Wastewater is a hotspot for resistance. Wastewater samples often contain especially diverse microbial communities and resistance genes. This does make sense. Wastewater collects microbes from around the hospital. Obviously, every time you wash your hands or tip water into the sink, you're likely introducing new microbial friends. Um, and this effectively acts as a summary of what is happening throughout the building. This finding reinforces the idea that hospital wastewater systems are critical reservoirs and potential pathways for AMR spread into the wider environment. Fifth and finally, evidence of shared risks across human and animal healthcare. Although the human and veterinary hospitals were distinct, the presence of overlapping resistance genes and mobile elements suggests that there are shared risks. This supports the idea that antimicrobial resistance should not be tackled in isolation within just human medicine. Instead, it does require coordinated approaches across human health, veterinary medicine, and environmental management. So we know what we found, but why does it matter? Antimicrobial resistance is often described as the silent pandemic, and it threatens routine medical procedures from surgery to cancer treatment by making infections harder to treat. Our study contributed to this global challenge by showing that hospitals, both human and veterinary, are important reservoirs of antimicrobial resistance, both when considered individually and also in a One Health sort of global context. The environment, especially wastewater, can be a reservoir for potentially harmful microbes as well as resistance genes. And using metagenomics and genomics in general can identify a lot of important factors for surveillance that other methods can't necessarily see. Combined, these insights are really important for helping to design better strategies to control antimicrobial resistance. Based on our findings, what can we do differently? There are several potential actions that stand out from what we found, including improving environmental monitoring. So, metagenomic sequencing provides a really powerful way to monitor AMR in real time across entire systems, not just individual pathogens. It strengthens a One Health approach. Policies and interventions should consider humans, animals, environment, agriculture, all these sectors together rather than separately. There should be a focus on wastewater and environmental pathways, and reducing the spread of resistance may require new approaches to managing hospital wastewater and environmental contamination. And finally, targeting mobile genetic elements. Understanding and limiting the spread of plasmids and other mobile DNA could be key to slowing down the spread of these resistance genes. All of these things are important, of course, but implementing metagenomics can be really expensive, and it also requires a lot of computing power to analyze the vast amounts of data that you get from it. Studies like this are really important for providing the evidence that policymakers use to decide whether it's worthwhile including these new technologies in their existing programs. Looking at the bigger picture, this study is part of a growing effort to use genomic technologies to better understand how AMR emerges and spreads. But looking beyond individual bacteria and examining entire microbial systems is much more sort of global. We can gain much richer and more actionable understanding of these different problems. Ultimately, tackling AMR will require a combination of better surveillance, smarter antibiotic use, improved infection control, and greater collaboration across disciplines. It will also require greater collaboration across countries as well. As a final thought, the comparison of human and animal hospitals highlights both the differences and the connections between these two environments. Whilst each had its own microbial fingerprint, they are linked through shared resistance genes and pathways of transmission. This reinforces a key message. Antimicrobial resistance is not just a clinical, so human or animal problem, it's also an ecological one. By understanding the full system in which resistance exists, we stand a much better chance of trying to control it.